Alrighty, welcome. My name's Calvin. This is the Bio 375 test, exam 2, actually. So I'm going to go through the problems really quick. Hopefully the class has done them already, so I don't have to explain too much in depth. But I'll give a little explanation of what we're looking for and how to solve it. So question number 1 is asking for the frequencies of the non-disease allele in the human population. This is with the given data of ADPK or ADPKD disease affects 1 in 100 or 1 in 750. Sorry, I'm not a video dude or presenter. So, with the data of 1 in 750 individuals affected by this disease, we're asked to find the frequency of the non-disease in the human population. And so that would be question A, if you can see that. So P or Q squared is the genotype frequency, and that is just 1 over 750. To find Q by itself, you take the square root of that, obviously just basic algebra, to get the 0.365. And then you subtract it from 1 to get your P value, which is 0.9635. Does that make sense? So you'd find the genotype frequency of the recessive, that is the affected, and then you convert it to the allele frequency, and then you subtract it from 1 because P plus Q equals 1. And so obviously if the recessive Q is this number, then P is just going to be this number subtracted from 1. And then question B is asking for the allele frequency of the recessive individuals. And so we just did this problem up here. And so our answer stops right here with 0.365. And then part C asks us for what the frequency of the heterozygous individuals in a population is. That is just 2pq right there. And so I did 2 times the allele frequency for the p value and also for the q value, multiplied those together and got 0.0703. Part D is out of the population of 1 million people, how many are heterozygous, or how many are expected? And so we take what we found in C and multiply by 1 million. And that's, I put it in scientific notation as 1 times 10 to the 6th, and times that by our heterozygous allele frequency right here, and we get 7,000 or 70,336 individuals are heterozygous for that allele, or for that disease. So that's question 1. Question 2 is a little more intuitive. It's asking us to identify the points on the graph. You can see I've already drawn on it a little bit to define where things are. So for the first one, W, you can see right, where is it, right there? That is a phosphate group, and those are all phosphates as well. For the X, which is above, there are sugars. In this case, it's a deoxyribose sugar, because we have the presence of the thymine. If it was ribose, it would be why can't I think of the name? Uracil. And then the next one is for the Y group. That is going to be this one right here. So adenine is paired with thymine. So you could argue that's a thymine group, or because adenine is a purine, it's paired with a pyrimidine, and so that could be also the pyrimidine group. Same question for Z. It's asking us what this one is, and then looking at this little pattern, it's got like an arrow instead of a block, and the arrow up here represents a G and a C, the G being the purine, the C being pyrimidine. And so in this case, you could say it's guanine, the purine, or just say the purine. Either one of those works. Part C, it wants us to identify the backbone, and I put a square around it. It's the phosphate group plus our sugars right there, along there, holding the nitrogenous bases in place. And then part D, I apologize for the poor quality of me back and forth paper. I don't know how to put a presenter up. But part D asks us for where the DNA molecule gets a net negative electrical charge, and that's coming from the bases in the middle there. They're charges with the hydrogens and sugars. Okay, so that's part 2. Question 3 is a little longer. It gives us a bunch of data, and we're asked to map it out on the gene, on the chromosome, and determine the order of the array, or the arrangement or the order of the genes that are linked. So I did it all on the back. So we start off with a heterozygous mother or parent, and then a homozygous parent or father in this case. Because of the information, we know that the mother is heterozygous and the father is homozygous, and these are fruit flies. So just really quick, K star represents the normal eyes. E star represents the normal body color, and then CD star represents normal eye color. And then regular K is ebony, or is bean-shaped eyes. Regular E is ebony body, and CD is cardinal eye color. Okay, so back to here, we see that different data. These are observed, right here, observed numbers for the population, how many had each phenotype, and it asks us again to find how those are linked and where. And so you start with your parent data, and you determine how a heterozygous mom and a homozygous dad could be set up. Then you take the parental offspring. Those are going to be the ones with the highest count in the population. And so in this case, it's this first one and then this second one. So those two genotypes are, genotypes are written here in the parental, and then you do the same thing with the double crossovers, and that's going to be the smallest one. So the 8 and the 6 right there. And then we list out 3 possible scenarios for what which one of these genes is going to be on the end here, so we can determine the order. And so our first scenario is just like the parental is set up. And so we're using the parental as kind of a template. So the first one follows the same template. The second one, we move the E genotypes to one end and squish the K over to the CD, and then we move the E to the other end on the third scenario. And then what we do is we do a little kind of dip where we capture this genotype, the regular K, this one regular E, and this one regular C, CD. And then we do the same thing where this one's E star, this one's K star, and this one's regular CD, and then K, and then regular CD, and then E star, if that makes sense. And so then we decide which one of these match our double crossover order. And so doing that, obviously, it's the second scenario that has E star on top with K and CD, and then E on the bottom with K star on the bottom and CD on the bottom. And so this is the order of the genotypes. And then we set it up. So this would be mom, how she is set up, and this was dad. And obviously she's still heterozygous and he's still homozygous. And then we come down here and we list out the same mom 4 times, and we take different groups. This is going to be from the distance between E to the K, I believe. Yep, E to K. And so we do for the genotypes for this offspring, so the offspring with all 3 of the stars, and then we do it for this offspring for none of the stars. And we follow that pattern just in reverse, or an opposite, flip side, flipped. So we start on the bottom, and we do 1, and then 2, and then 1 on the top, and then 2 on the bottom. Just kind of reciprocals of each other. And that gives us the rest of the offspring as observed here. And you'll notice if you go through that each of these corresponding phenotypes are matched with what we just found. So in this example, 98 individuals right here had this genotype, and the same for 97, and so on. Then we add those together in groups. So this first group where we did the distance between E and K, we added that to the double crossovers. And so the 8 and the 6 again were our double crossovers. Put those in the equations plus these 2 population size, well, sample sizes from the observed data, and we divide it by the total amount of the population. So there were 4,000 all added up, and then we multiply it by 100 to get a percentage, and that's 7%. Do the same thing on the bottom, and that's 5%. So now we take this information and put it on the map. So we said from E to K, there's 7 units, MU, map units. And then from CD to K, there are 5 units. Okay, so pretty simple. And then to calculate interference, this is how much it should have happened based on what didn't happen. So we take the map units divided by 100 to get it back to decimal form, multiply them together, and then we multiply it by 100 again to get a percentage. Then we multiply that percent by our total population to get, this is how much of the population actually had were recombinant. So we divide that by 14, which is our double crossovers, and to give us the 1% right there. And this is how often it actually, or it didn't express itself. And so you subtract it from 1, 99, or no, it did express itself. It didn't express itself. Subtract it from 1, and so this is how much we should have observed, but we didn't. If I understood the material correctly. So that's it for number 3. And then finally, number 4, it's basically a repeat of number 2. It's asking us to find the frequencies of the D allele, the recessive D allele, and then the homozygous, or the recessive and then the dominant alleles. And then it asks for the frequency of the DD genotype and also the D little d. So the first one was big D, and then the second one was little d genotypes. So we're looking for genotypes instead. So we'll do something a little different. And so we're given a data value of 1 over 32,000 is are the individuals affected, so they are recessive. So that is PQ, or Q squared. And we take the square root of that to get this value right here. And then part B, we subtract it from 1 to get our p-value. Part C, what I did, and I'm not sure if this is correct, but what I did is I took the Oh, and I also solved for the heterozygous, so that's right here. So 2PQ. I figured that would be information to know when finding genotypes, because these heterozygotes also contain some of the dominant alleles and also some of the recessive alleles. And so I multiplied what I found for the heterozygous number, which was 0.0112, and I added it to our p-value for the dominant, and I squared our p-value first to get 0.99997 is the genotype of the population. And then for part D, I just squared our heterozygous group to get the genotypes. Again, genotypes is squared. And that's it. So if you have any questions, please ask the teacher, because she knows a lot more than I do. But that's the work I did, and I hope that made sense. Thank you for watching.